Assalamualaikum and good morning, Doctor. Today, Group 5 will present video lab report, blood collection and smear preparations in fish. Before we start our presentation, I want to introduce my member in Group 5. Number 1 is me, Elena binti Jinuddin. Number 2 is Siti Norada binti Isnil. Number 3 is Daniel Phillips. Number 4 is Onicha Evi. Number 5 is Maswe Martin. And number 6 is Christina Libia Suntai Anak Emai.
Begin with, I will give a brief introduction about this lab report. So the blood is a fluid that is vital to all animals, including us humans. They are used to regulate our temperature, to transport nutrients, oxygen, carbon dioxide throughout our body, and most importantly, they are important to protect ourselves against the infection of diseases. So in this experiment, we collected blood from a live fish sample via syringe, and we can observe it under a microscope.
Hello, my name is Maswe Martin and I will be showing the objective in this experiment. The experiment of blood collection and smear preparation in fish. First objective is to learn the technique of blood collection from fish. This means we will be learning the right angle to take a blood fish and where the perfect location on a fish to take the blood from. It also includes the necessary tools and protocol that we can follow in order to correctly take blood from fish. Second is to prepare blood smeared for microscopic examination, enabling the identification of blood cells and understanding their function. Again, knowing the right tools it will be helpful, and this objective will be useful in practical use. That is all from me. Thank you very much for listening.
Hello, I'm Siti Norada binti Isnil, BS 221605551. Today I will present about materials needed on blood collection. The first is live fish anesthetic solution MS222, sterile PBS, stand for phosphate buffer saline, sterile syringes with 1 ml or appropriate size, sterile needles 23 until 25 gauge or appropriate size, and anticoagulant, for example heparin, sodium citrate, EDTA, and microscope slide and cover slip. Also tubes, hemocytometer, microscope, right stain, origin stain, and disposable glove and lab coats. That's all.
Methodology for blood collection is before starting the experiment, we have prepared a clean and strict wall area. Second, the antiseptic fish to remove from the aseptic solution and place on a soft damp cloth to prevent damage to the fish. We need to find the tail, vein or dorsal aorta, which are commonly used site of blood collection in fish. And then we use strings and needles carefully puncture the vein or aorta and slowly draw blood into the string. Make sure the collected value is sufficient for the desired analysis. And last, once the blood has collected, we remove the needle and remove the blood into a clean, labeled microcenterized and or a Pandolf tube. Methodology for blood smear and staining. First, before placing the accumulated blood, we clean the microscope slide with alcohol and let it dry. Second, then we put a small drop of collected blood near 1/8 of the slide. After that, we use a second microscope slide head and at an angle of 45 degrees, spread drop of blood among the first slide to produce thin smooth. 4. Let the application dry completely. We have prepared a solution of bright dry coloring or ginseng coloring according to the manufacturer's coloring. instruction. Then we put a dry and fixed smear of blood with methanol 1 to 2 minutes on the dry rack or dry plate. After placing the blood, we add enough ginseng dye solution to cover the smear and leave it sweet stable for the recommended coloring time is 2 to 50 minutes. After 50 minutes and the blood was as dry, we rest the slide gently with distilled water to remove acid and we let the slide dry completely. Once dry, the cover has been placed over the stain smears. Using mounting medium is necessary, avoid air bubble. After that, we examine staining smear under a microscope at various magnification to observe and identify different types of blood cells, for example, red blood cell, white blood cell, and observe their characteristics.
Assalamualaikum Doctor, I'm Elena binti Janet Lim going to explain the result in this lab report too. The upload photo show in the result blood collections from fish under a microscope. The image appear to display a decent concentrations of cells which are likely red blood cells based on their uniform appearance and distribution. Round shape and relatively consistent cell size are typically characteristic of erythrocytes red blood cell in many fish species. The current image provides a qualitative view showing successful blood collections with a good concentrations of cell that can be seen under the microscope. That is for me. Thank you.
My name is Anisha and we make this by SQ21603. In the last slide, I'm going to talk about the conclusion. Our observation highlights the critical importance of minimizing stress during the blood collection. We found that a proper handling, effective anesthesia, and efficient blood withdrawal were all crucial in reducing stress-induced variation in hematological parameters. Accurate interpretation of fish health relies heavily on obtaining blood samples that truly reflect the physiological state of the fish. Furthermore, we gain proficiency in preparing a high-quality blood smears, recognizing the importance of a smooth, feather edge and a monolayer of evenly distributed cells. Rapid drying techniques were practiced to prevent cell distortion, and a proper methanol fixation was applied to ensure optimal preservation of cell morphology for accurate microscopic examination. In conclusion, this laboratory session equipped us with the fundamental skill necessary to collect and process fish blood samples effectively. Thus, we have successfully achieved our objective.
That is from video lab report too. Thank you.